Hello, and welcome to our webinar on a simple single-tube approach to processing sexual assault samples. I'm Leanne Landers. I'm the Director of Strategic Marketing here at MicroGEM, and I'm with our speaker, Jeff Hickey. And we are thrilled to welcome our colleagues, collaborators, and people from across the forensic community who are joining us today from across the U.S. and even from across the world. Jeff is our forensic expert here at MicroGEM, where he leads business development efforts in the United States and provides technical and product development expertise. Before joining MicroGEM, Jeff worked in human ID markets, both with the nuclear DNA section at AFDIL and with Cellmark Diagnostics, where he performed DNA analysis and body fluid analysis. He received his Master of Science degree in biotech from Johns Hopkins. So today we're talking about a rapid approach to DNA extraction using heat and enzymes. It doesn't need chemicals and multiple wash steps. It preserves DNA from your precious samples. It drastically reduces hazardous and plastic waste and limits contamination and human error. And it does all of this in minutes, not hours. Jeff will talk about this approach and how it's being successfully applied to forensic samples, including Y screening and differential extractions. We'll leave time before the end of our hour together for Q&A, so please feel free to submit your questions as we move along. Just use the Q&A button at the bottom of your screen. And please feel free to chat if you want to communicate with us as well. So with that, I turn it over to Jeff.
Thank you, Leanne. So just to get started here, I was going to talk a little bit about sort of the history of differential extraction and what we're doing differently than what was done in the past. And seeing this slide here where, I know that there's people on this call, probably plenty of people who when they started in forensics, they did RFLP, like you're looking at that audio radiograph there, which is what we were doing when I started in forensics back in the 90s. And, it's particularly poignant to me in this call because a lot of the work that I'm presenting here has been done in collaboration with Robin Cotton at Boston University. And she's unfortunately not able to be on this call right now, but she was my first lab director at the first forensic lab where I worked, Cellmark Diagnostics. And back then we were doing differential extractions exactly this way. And just a little aside story that amuses me, and that maybe with a webinar it's a little odd to not get feedback on this, but basically I have a recollection with Robin that I started there as a lab assistant, and when it was time to become a forensic examiner and I was applying for the job, Robin called me into her office and said, I know that you can do the science. I think you'll be great at that. She's like, I just have one question for you, really. And she says, do you own a suit? And because I guess I was dressed very casually for work all the time as a lab assistant. And so I did indeed get a suit. And in fact, the first forensic case I ever did ended up going to court. So Robin and I have stayed in touch throughout the years. And recently we've been collaborating on these projects, and it was work from her lab that we were able to commercialize. So looking at differential extraction history, going back to the old days, differentials have been done pretty much the same way, which is to start by lysing the epithelial cells in a cocktail of detergents and proteinase K. And once you've lysed the epithelial cells, you're going to do a bunch of centrifugation steps, usually, and washes, although sometimes people will use a DNase at that step instead of doing centrifugation. And then as you move on after those centrifugation steps and pelleting your sperm, people typically lyse the sperm with DTT, proteinase K, and detergents. And of course, all of those things together would be very toxic to PCR. So you're going to be doing all these purification steps after that, which adds time, cost, and of course you're losing DNA along the way. So when we look at where we've progressed in forensics, obviously people are not doing RFLP anymore. Actually, if anyone out there is doing RFLP, please send in a chat because I would love to hear about that. But I don't think very many people are out there using RFLP in forensics. But people are still generally doing differential extraction the same way. I mean, there are people out there, obviously, who've thrown in a DNA step instead of doing the wash steps for the pellet. And of course, there have been commercial kits that have come along instead of doing phenol chloroform extractions. But largely, a lot of this has been the same since the late 90s or so, and the only real advances have often been just in automating processes, but not actually changing what you're doing chemically. So what we're looking at is just dramatically changing the actual chemistry to eliminate a lot of waste along the way. And so when you look at the ways that people currently extract DNA often and the ways they did in the past, of course, back in those old days of RFLP, everyone was doing the solvent extraction, the phenol chloroform extraction methods was what people pretty much did. And so in the case of digesting sperm, you would need proteinase K and SDS and DTT in large amounts to lyse the sperm. And then you're going to be using phenol to remove all that chemistry that you put in that's going to be kind of incompatible with your downstream assays. And then you're going to use chloroform to remove the phenol, and then you're going to use ethanol to remove the chloroform. And then you're going to have to centrifuge and wash with more ethanol and let it evaporate. And all these steps along the way are places where you could switch your samples, you could have contamination, and more importantly, you could also be losing your sperm DNA along the way. Now, there have been a number of commercial kits put out that use columns or magnetic beads instead of the phenol chloroform, and some of these can be automated. But largely, you're still starting at the beginning with that DTT and the SDS and the proteinase K, only now you're also adding guanidinium HCl, which is toxic to PCR. So you're going to then do a lot of steps washing over magnetic beads, and it's well known that you lose DNA throughout this process. So if you're dealing with samples with very few numbers of sperm, you're definitely going to be throwing away some of that sperm along the way. And I'll show some data later on that shows just how much you're losing. So some of the drawbacks for these traditional DNA extraction methods, obviously I mentioned contamination. So it's all these points where you're opening the tube and adding reagents along the way, or perhaps even transferring samples. If you're doing these manually, this is also just a lot of labor. If you've automated it, typically automating these processes, when you're doing columns or magnetic beads, you may need some kind of custom automation in order to do it. And so then you're kind of stuck with a specific consumable, which is going to usually add cost and waste along the way. So now that I've sort of talked about how people are kind of doing it, I want to talk about, how is it that we're able to avoid those steps at MicroGEM? And the way we're able to avoid those steps is that we have a novel proteinase, which operates at a much higher temperature than proteinase K. It operates at 75 degrees Celsius. And the reason why that's so important is that we really just don't need to add in all of those detergents to assist our proteinase in denaturing the proteins. They're already starting to denature at those temperatures. And so this enzyme has quite a bit stronger activity than, say, proteinase K. The other interesting thing that's important to our sperm lysis chemistry is that it has very low activity below 50 degrees Celsius. And it can be killed with a 95 degrees Celsius heat step, so it doesn't interfere with PCR amplification. So in general, our protocols, when you look at just sort of our generic chemistry, what we might call our universal chemistry, if you're dealing with epithelial cells, for instance, with our chemistry, you would simply go to 75 degrees Celsius using our buffers and our enzyme. And at 75 degrees Celsius, our proteinase is going to become active, the cells will lyse, we're going to destroy any nucleases, and we're going to strip the DNA of proteins that would interfere with PCR. When we shift to 95 degrees Celsius, we're going to deactivate our proteinase. So really just a few minutes at 75 degrees Celsius and then a few minutes at 95 degrees Celsius, and you have DNA that's ready for PCR. Now, what we've discovered in recent years, down at our lab in New Zealand, they've done work with bacteria and plants, and they've found cocktails of enzymes that can be combined with this proteinase in the same reaction. And in that way, we can get even basically combining processes that are not normally combined in the laboratory. And that's due to the low activity of our enzyme at these lower temperatures. So for the example of lysozyme, which is often used in bacterial extractions, we can add the lysozyme at the same time as the proteinase and simply in a thermal cycler sit at 37 degrees Celsius where lysozyme is active, go to 75 degrees Celsius where our proteinase is active and destroy the lysozyme, and then at 95 degrees Celsius, we're going to destroy our enzyme. What was discovered in Robin Cotton's lab and what we've commercialized, she found a cocktail of enzymes that are active at 52 degrees Celsius that work in concert with our proteinase and specifically for lysing sperm. So we're not using DTT, we're not using other reduction agents that people have used in the past for lysing sperm. This is an entirely enzymatic process, which is why we don't need all the downstream purification. So the first application that we found for this sperm lysis chemistry was actually, we were at an ISHI conference a few years back, and I had a number of labs come to me and ask if we had a solution for Y screening because they knew about our rapid enzymatic chemistry. And at the time, we hadn't quite commercialized the sperm lysis kit, but it was interesting, the interest in Y screening. And so we did commercialize it and have worked with some labs to validate it and also did some internal validation work. But this is, in our quick start guide, we have just a basic protocol for semen stains and liquid semen. And the standard protocol for us is a 100 microliter volume. So 10 microliters of our Orange Plus buffer, which is, it's sold at a 10x, and you're essentially diluting to a 1x. You use 2 microliters of our forensic gem, which is the thermophilic enzyme that I was talking about. 10 microliters of Acrosolve. Acrosolve is the cocktail of mesophilic enzymes. And add water up to 100 microliters. And this can be done, then run in a thermal cycler, or you could use multiple heat blocks. You could also use our PDQX instrument, which I'll show in a second. But this can also be scaled. So while we've done a 100 microliter reaction here, if you find that you want to work with a larger piece of material, that can be scaled up. If you want to work smaller, that can be scaled down, which would also obviously have cost savings since you would be using less than a full reaction. So 100 microliters can be moved around. As well, these temperatures and times, I would not change the temperatures, but the times can be validated. So some customers could validate longer than 5 minutes at 52 degrees Celsius if they found that aided in the extraction. So this is kind of the basic protocol, the basic thermal cycler workflow. You could pre-dispense the reagents into a 96-well plate and then take cuttings. In the case of Y screening, you could take cuttings from, say, vaginal swabs as a way to do the screening instead of body fluid analysis or in addition to body fluid analysis. Or you could do this vice versa, do the sample cutting first and then dispense in the master mix. And then really any thermal cycler can be used. In this case, we have an older 9700 in our lab, so that's what we use. But just 5 minutes at 52 degrees Celsius, 3 minutes at 75, and 5 minutes at 95 degrees Celsius is sufficient to lyse the sperm and the epithelial cells. So this can then go on to real-time PCR. And actually, though, it can go on to STR amplification as well. So if some labs prefer to work with our PDQX nucleic acid extractor, and so the PDQX is an alternative to the thermal cycler. It does come with a consumable tube, which can hold up to about 150 microliters of liquid, and it has a very small footprint, so that's why this pen is here to kind of show you the approximate size of the instrument. It runs 24 samples at a time, and it's quite small. So some of the studies we did related to Y screening, we of course did a sensitivity study where we wanted to see, can we detect really low levels of sperm cells in an abundance of female epithelial cells? And so we had some known female epithelial cells, and we had some sperm some semen that we knew the sperm count. And we just diluted out the sperm cells in the epithelial cells to get down to where we were down to only one sperm cell per microliter, which is pretty much the limit of detection of the real-time PCR kit. Anyway, in this case, we were using the Promega Plexor HY chemistry. So we ran these samples, and looking at the average of the samples, when you're down there at one sperm per microliter, which to me is probably the most important of these of this dilution series, you're definitely down there detecting one sperm. And we didn't find any instances. We actually did a number of different variations on this study. We never found an instance where we didn't detect that one sperm per microliter. So moving on from that, we wanted to see, okay, well, does that real-time PCR data actually correlate well with what we see in STR amplification? So we took the samples that had about equal portions of sperm and epithelial cells and amplified that. And here's an example here, and it looks like it's about 50-50 male and female, which is what you would expect. Down here, the sperm, the one that had 50 sperm per microliter in about 370 epithelial cells per microliter, you can see that you're just kind of barely seeing the male fraction in that mixture. And so that's kind of what you would expect at that ratio of sperm to epithelial cells. And of course, the other ones would have been too dilute to detect the male at all. But basically gave us the idea that, yes, this has relevance to the downstream processing. So we made some mock casework samples by making a similar dilution series, but depositing it onto cotton swabs and allowing it to dry. And these samples were run, and similarly, we were able to extract off of dried swabs. So to make some more mock casework, many times people talk about denim and they talk about soil as being potential inhibitors for PCR, for STR amplification. So we had some dirty genes that we deposited some semen sperm count on these genes, and on purpose these important, and we stored them at room temperature for a month and then extracted them. And of course these worked fine, so we got great STR profiles off of those. This was using the Promega PowerPlex Fusion kit. So really showing you didn't necessarily need all that purification to get rid of the denim or the soil inhibitors. So this is after kind of doing some of these initial tests. We've gotten interest from some people, and in particular, back in 2017, the New York Office of the Chief Medical Examiner presented their Y screening validation of this chemistry at the American Academy of Forensic Sciences meeting in New Orleans. They presented that, and they've implemented it into their casework. They have their protocols available online, so anyone can easily see what they do. We've also had DNA Solutions presented a poster at the last ISHI conference, the ISHI 2019, and that was on their validation of this chemistry for Y screening as well. And so we have a couple using it for Y screening, and I know that they have data taking it on to STR amplification. And so the kit is very much compatible with both real-time PCR and STR amplification. And then we've had some publications recently, particularly James Lander's lab has put out a few publications. One was using it for Y screening using a novel LAMP assay. And there was also one using acoustic trapping of sperm cells. And there's also another of other universities out there using it for research that I don't have on here because I haven't seen their publications yet. But I think a lot of people see the value in this for microfluidics, for anyone trying to implement it into an automated process because it doesn't have the purification steps. So moving along, we wanted to do a good comparison of this to a commercial competitor. And so we actually looked at two competitors, but initially I'll show this first competitor first. We wanted to mimic what it would be like if you did a differential extraction and now you have your pelleted sperm. And really prove that you're losing the DNA by using these other technologies, by using columns and magnetic beads. And so in this case, I took some liquid semen and diluted it 1 to 10, 1 to 100, 1 to 1,000, and 1 to 10,000. And I made 6 replicates, 6 100 microliter replicates at each of those dilutions. And then I spun those down in the centrifuge for 5 minutes to pellet the sperm at the bottom. And I removed 80 microliters of the supernatant and discarded it, so that really you had a sperm pellet sitting in 20 microliters. And so this, I thought, would be a fair way to compare and say, okay, we'll follow our method and extract, and we'll use the competitor method, and we will extract and see who's doing better on the extraction. In this case, the advantage of the MicroGEM reagents is that we can work in really small volumes. And so because this is only 20 microliters of sperm here, I don't need to extract 100 microliters. I can simply scale our reaction, sort of pretending that this 20 microliters is just water, and add 3 microliters of water, 3 microliters of Acrosol, 3 microliters of Orange Plus buffer, and 0.7 of our forensic gem enzyme. So I'm only working in a 30 microliter reaction. For the competitor's method, I followed their liquid semen extraction method from sperm pellets. So looking at the quant data, we ran the real-time PCR, and you'll see MicroGEM is the dark blue, the competitor is in gray, and this is nanograms per microliter. And so at a 1 to 10 dilution, of course, MicroGEM is off the charts, getting more DNA than the competitor. And as you go down, that stays consistent. But down at 1 to 10,000, you can see that you don't actually get any quantifiable DNA from the competitor kit, whereas we're still getting enough for SDR amplification from the MicroGEM kit. So I calculated the percent loss at those dilutions by looking at the competitor chemistry and subtracting what they're losing compared to what we got. And they're really losing anywhere from 85% to about 95% of the DNA in these samples, which is pretty important for forensics. So looking at this, sometimes people say, okay, but that's just kind of a dirty extract, or that's quick and dirty, and it's not really true. This is a gorgeous STR profile that we got from that 1 to 1,000 STR, from that 1 to 1,000 sperm sample. And this is because we're purposely tailoring our chemistry so that it's compatible with these STR kits. It's not simply a crude extract. So looking at heterozygote peak balance, because that is another quality metric that forensic labs look at a lot of the time. MicroGEM is in dark blue, the competitor kit is in gray, and you can see that across the board, MicroGEM is performing excellent in heterozygote peak balance. I know there are some non-forensic people on this call, so I would just say that being close to 1 is where you want to be when you're looking at this, and we're clearly doing quite well. So really looking at the 1 to 10,000 dilution here, you can see the competitor has 0 peaks that are above 100 RFUs for this sample, whereas the MicroGEM kit is actually getting almost a full profile in this case. So still getting useful data all the way out to there. And so we looked at another competitor kit. This was designed exactly the same way, this particular experiment. We just did a slightly different dilution series, 1 to 500, 1 to 1,000, 1 to 2,000, 1 to 10,000, and calculated the percent loss. And this other competitor kit, and these are both 2 commonly used kits, they're also losing anywhere from 75-ish% of the DNA in the sample to 90-something% of the DNA in the sample. And if we look just at the straight quants in this case, MicroGEM is in orange and the competitor is in blue, and it's basically the same that we were seeing with the other kit, where MicroGEM is getting dramatically more DNA from these samples. We did not amplify that one for STRs, just went with the quant data for now. So getting back to work we've been doing with Robin Cotton, she has moved on beyond this, of course, Y screening, and has had students working on creating a differential method, because she found the value of our high temperature proteinase, she found some other application there, where she found a nuclease called benzonase, which could also be used in conjunction with our proteinase. So rather than the traditional extraction method, she's going to do an epithelial cell lysis using our ForensicGEM universal kit. And so this is a very rapid epithelial cell lysis. So we go to 75 degrees Celsius for 15 minutes. Then she's going to centrifuge down to pellet the sperm and then remove the supernatant from that. And now she's going to add a cocktail of benzonase with some more of our ForensicGEM enzyme. And the benzonase is active at 37 degrees Celsius. So if you recall, our ForensicGEM enzyme is not going to have activity at that temperature. Benzonase will chew up any residual epithelial cell DNA in the sample while leaving behind the sperm pellet intact. And then at 75 degrees Celsius, when we ramp to that temperature, the ForensicGEM enzyme will destroy the benzonase. So this is a very efficient way to shut down that nuclease. I know that other people have used other DNases. Sometimes they need to be shut down chemically, sometimes with temperature, but this is a very effective combination. And then we lyse the sperm using that sperm lysis chemistry that I just described. And so now we've also just removed, if we were doing this with traditional methods, you would still have to clean up this epithelial cell fraction, and you would still have to clean up this sperm cell fraction, which would obviously add a lot of time and cost to the sample. So this is just sort of a general description of Robin's protocol, and this was presented by her student Mike Yaku at the ISHI conference a couple of years ago, and she allowed me to use these slides. But yeah, benzonase is not made by us. Benzonase is made by Merck. You can purchase it from Millipore. It's non-specific, so it's going to digest both double and single-stranded nucleic acids. And so we can use this to digest any residual E cell DNA in the sperm fraction. Optimal activity at 37, and it can be degraded by the EA1, the ForensicGEM proteinase. So it can be used in sequence. And then after that, we can add in the sperm chemistry, and I'll have a better description of her protocol a little later on. But so this is data that she presented. She and her student Mike presented a couple years ago, which was just showing they did a traditional differential extraction with spin steps and washes like you would normally do. And you can see these highlighted peaks are the male fraction or the female fraction coming into the male fraction. And so it didn't have a very good separation. When she did the same samples using her benzonase method, you're getting a clean male fraction, and importantly, you're also not seeing degradation of the loci with the benzonase. So at the time, they presented this as sort of, they had done a few studies, and they were working with small samples in a thermal cycler. They were taking very small swab cuttings and working in a 100 microliter volume at the time. So one of the things that she had wanted to work on was working with larger swabs, because a lot of times you may need to take half of a swab or something like that. So she kind of had to do some optimization on the chemistry with her students, and then making sure that they had optimized the benzonase to remove the male DNA but not interfere downstream, and make sure that our ForensicGEM enzyme was fully shutting down that benzonase. So this work was done using the Thermo Fisher, the Identifiler kit, or it may have been Identifiler Plus kit. And so that was with her student Mike Yaku. Now she had another student come along named Andrew Ziegler, and he created this data. And the unfortunate thing that we discovered when she switched to using Global Filer, she started observing what we were losing some loci. And so she did some studies to sort of isolate the problem, and we discovered by doing dilution series that our Orange Plus buffer was actually inhibiting Global Filer. And I would say that all the testing we had done prior in-house was using Promega chemistry. partially because our early customers that were interested were using Promega. So looking at that, we had Robin try out some of our other buffers because we make a number of different buffers. Looked back at some buffers and discovered that we actually have a different buffer that is compatible and that is compatible with Global Filer. So her student Emily Hoffman created this data. And so now, once she was able to realize that it was really just that particular buffer and that we had another available buffer that we could solve the problem, they were able to proceed with their testing. So I'll briefly describe her protocol here, and there will be more detail, obviously. If people are interested in trying this out, we can certainly get the fully detailed protocol out to people. Robin is willing to share this with people. And so basically, it's taking a sterile scalpel. They've been working with about 1/4 of a swab head. They place the swab head into a tube that they label sperm fraction, and they're going to add these reagents in that first step. So this is our MicroGEM universal kit, essentially, only she's scaling up to a 500 microliter reaction rather than 100 microliter reaction, so that she can fully submerge that piece of material. And then you're going to do 75 degrees Celsius for 15 minutes. And so we're lysing the epithelial cells at this temperature, but our MicroGEM enzyme without Acrosolve will not lyse the sperm cells. So at 4 degrees, we're going to go down to 4 degrees Celsius, and that's actually an important step. We're not just putting that in there as a hold. It's important to go down to 4 degrees Celsius because when we add the benzonase, we want to make sure that that MicroGEM enzyme is not active. So it needs to be cooled down to 4 degrees. So then she's going to take tweezers, and you're going to remove your substrate material. And you could, at this point, put it in a spin basket like you would with any method. Robin is actually doing some research around taking it out and saving that material and calling that the material fraction, because there's still going to be viable sperm, well, not viable sperm that could be intact for lysis. There's going to be sperm DNA in there that could be valuable evidence. So she actually has in her protocol to continue testing on that, what she's calling the material fraction. But you could certainly use a spin basket like you normally would. In this case, you're going to lose some of the liquid to that swab. So she was estimating 50 to 60 microliters would be lost to the swab. And then she would treat that swab material with benzonase and MicroGEM cocktail in order to get rid of any epithelial cell DNA that's still on the swab. Now, in terms of the sperm fraction, so now you remember we've removed the material, but we now still have the volume of liquid, probably 400 microliters or so, that has sperm cells intact and epithelial cells that have been lysed. We're going to spin that down like you would in a normal differential extraction, and we're going to remove 400 microliters of the supernatant and transfer it to a tube labeled E-cell fraction. So these are your epithelial cell fraction. Unlike other chemistries, that is now ready to go for real-time PCR or STR amplification. You don't need to do any purification steps at this point. So that's definitely going to cut down on time. At this point, she would add 12.5 microliters of her benzonase reagents to the sperm pellet. And so we have the recipe here. It's 10 microliters of 2.5 units per microliter of benzonase, which has been already pre-diluted in a 2x benzonase buffer that's supplied with the benzonase enzyme. And then she's going to add to that some diluted MicroGEM enzyme, which has been diluted in our blue buffer. And so she and her students spent quite a bit of time really working out how to make these buffers compatible so that the enzymes functioned correctly. And so now this can go into a thermal cycler and go to 37 degrees Celsius for 15 minutes where the benzonase will be active. And when you ramp to 75 degrees Celsius for 15 minutes, the MicroGEM is going to chew up the benzonase. And then you're going to go to 4 degrees Celsius for 5 minutes. And you need this, once again, this 4 degrees step is actually very important. It's not just a hold. That is because we want to be at a cool temperature when we add in our sperm lysis reagents. You don't want the forensic gem enzyme to chew up your Acrosolve enzymes. So after that benzonase treatment, you don't need to do a spin step or anything. We're going to add 9.5 microliters of water, 1 microliter of 10x red buffer, if you're working with global filer in particular, and 5 microliters of Acrosolve, which is our sperm lysis reagent. And so now we want to ramp to 52 degrees Celsius for 5 minutes. That's where Acrosolve will be now active. And at 75 degrees Celsius, our forensic gem enzyme is going to chew up the Acrosolve so it doesn't interfere with any downstream processes. And at 95, we're going to deactivate the forensic gem enzyme. And then we'll go down to 4 degrees Celsius, and that's really just cool the samples down for further processing. And so we just have a quick example from Robin. So she now currently in her laboratory is working on some real post-coital samples. She has an IRB through Boston University. And so now we're not trying to do so much mock samples anymore as we are trying to work on real post-coital samples. And so she's still in the middle of analyzing all that data and has her student Emily who's working on that, Emily Hoffman. But they did share with me just a quick example. So here we have an epithelial cell fraction that was generated using this method. And then here's a sperm fraction, and we're just showing, this particular color channel so you can see amylogenin very clearly. But basically, we're getting very clean sperm fractions from these postcoital samples and nice balance, even though we're not doing any purification steps. And so, yeah, her work is ongoing, and hopefully, one of these days, maybe we can get Robin to present it when it's fully completed all of the postcoital samples. So really, that kind of brings me to the end of the data that we have to share today. And I've had a lot of people ask me at conferences. People have been aware of the work that Robin is doing in her lab. And so I wanted to share with people those protocols because people ask for it. I would say that if anyone's interested in doing this particular method, trying this differential extraction method, they can contact me at j.hickey@microgembio.com. Just thank you for listening, and yes, I'm happy to help anyone out who wants to try and implement this in their lab, whether it's the Y screening or the differential, or if you're interested in trying out the universal kit for reference samples.